there is a conversion in defining two strands of DNA, okay, and the structural gene of a transcription unit. Since the two strands of have opposite polarity, that is 3 to 5 and 5 to 3 and the DNA dependent DNA dependent RNA polymerase also catalyze the polymerization in only one direction. The catalyzation process only occurs in one direction. So that is 5 to 3 n direction. The strand that has the polarity, that is 3 to 5, act as a template during this process, okay, and it is also referred to the template strand that is required for the copying of genetic information. The other strand has the polarity as 5 to 3 n and the sequence same as RNA except thymine at the place of uracil is displaced during transcription. And strangely, this strand which does not code for anything is referred as a coding strand. It will not code for anything, so hence it is called a coding strand, okay. And during this process, at the reference point, while defining a transcription unit, it is made with the coding strand. To explain the point, coding strand, a hypothetical sequence from transcription unit is represented below. Let me show you the transcription unit from the 3 to 5 n direction of the particular structure or template strand, and 5 to 3 n direction which indicates a coding strand. 5 to 3 n direction or 5 to 3 n strand is nothing but a coding strand which will not code for anything further. You can see here the transcription unit, okay, that consisted of promoter, structural gene, terminator, okay. Here you can see the transcription started site from the promoter region where genetic information is being copied, and apart from this, the promoter and terminator flank the structural gene, okay. The promoter and terminator flank the structural gene, objective from structural gene in a transcription unit. The promoter is said to be located towards a phi end of structural gene, okay, and whereas the next, it is a DNA sequence that provides binding site for RNA polymerase, okay, and it is the presence of promoter in a transcription unit also defines the template and coding strands. By switching its position with the terminator, if you carefully see the terminator, the definition of coding and template strand would be reversed in case of a terminator, and the terminator is located towards a 3 end or downstream of the coding strand, and it is usually defines the end of the process of transcription. This is the end of transcription, which means copying of genetic material was done, that is at the downstream process at the 3 end. There are additional regulatory sequences that may be present further upstream or downstream promoter. Some of the properties of these sequences shall be discussed while dealing with the regulation of gene expression, okay. Remaining part we will discuss in the next class. Until, stay tuned. Thank you. Have a nice day.